Today, we're back with Unintended Consequences, episode 39, for June 24th, 2026. Every story we cover starts with someone who had a reasonable plan. In the spring of 1968, a Boston gynecologist examined a cluster of young women, some still in their teens. The patients arrived with a diagnosis of clear cell adenocarcinoma of the vagina, a form of cancer that develops in the tissue lining the vaginal canal. That particular cancer was so uncommon in their age group that the hospital's pathology department had seen only one prior case in 30 years. Each of the patient's mothers had taken the same medication during pregnancy. The medication was diethylstilbestrol, or DES. The drug had been introduced with the explicit goal of supporting pregnancies that might otherwise end in miscarriage. Instead, it produced a cancer that would not appear until the exposed daughters reached adolescence or early adulthood. The compound was first synthesized in 1938 by British biochemist Sir Charles Dodds and his team at the Courtauld Institute. Dodds was seeking an inexpensive, orally active estrogen that could treat menopausal symptoms without relying on scarce natural extracts from animal ovaries. Within a few years, American obstetricians began testing the same molecule for a different purpose. They wanted to address recurrent miscarriage and preterm labor. At the time, miscarriage was understood largely as a failure of hormonal support. Estrogen levels were observed to drop before many spontaneous abortions. Researchers at Harvard and the University of Chicago therefore reasoned that administering a potent synthetic estrogen might restore the hormonal environment required to maintain pregnancy. Early case series published in the American Journal of Obstetrics and Gynecology appeared to show reduced rates of loss. The logic seemed sound given the limited tools available to physicians in the 1940s. By 1947, the FDA had already approved DES for menopausal symptoms. Its use in pregnancies spread rapidly through off-label prescribing. Pharmaceutical companies marketed it under more than 200 brand names. By the mid-1950s, an estimated 5 to 10 million women worldwide had received the drug during gestation. That range, 5 million on the lower end and 10 million on the higher end, reflects the rough counts compiled later from prescription records and marketing data across multiple countries. Professional journals carried advertisements claiming it produced stronger, healthier babies. A small number of researchers, including British pharmacologist Sir Robert Robinson, had noted in the early 1940s that high-dose estrogens could alter genital tract development in animal embryos. Yet these findings received little clinical attention. Most practitioners viewed DES as a low-risk intervention that addressed a common and distressing problem. One might ask why the animal data did not slow adoption, but at the time the prevailing view held that findings in non-human embryos did not necessarily translate to human pregnancy outcomes under medical supervision. In 1971, physicians Arthur Herbst and Robert Scully published a case control study in the New England Journal of Medicine. The study linked maternal DES use to the rare vaginal cancers appearing in their daughters. The median age at diagnosis was 19. Subsequent research established that DES crossed the placenta and interfered with the developing miliarian ducts. This interference produced permanent structural changes that later became malignant. The same exposure also increased risks of infertility, ectopic pregnancy, and breast cancer in the daughters. Later studies documented elevated rates of hypospadias and other genital anomalies in sons. Because the drug had been given to healthy pregnant women rather than to patients already ill, the population exposed in utero was unusually large. The latency period of roughly 20 years meant that the connection between prescription and disease was invisible to the original prescribers and to the women who took the medication. No short-term clinical trial could have detected an outcome that manifested only after the children reached adulthood. On 22 November 1971, the FDA withdrew approval of DES for pregnancy use. Cancer registries and specialized clinics were established to follow exposed individuals. Several large lawsuits reached settlement in the 1980s. The drug remained available for other indications for a time, but its obstetric use ended abruptly. Long-term follow-up has continued into the third generation. Some grandchildren of exposed women show subtle reproductive tract changes, although cancer risk in that cohort remains under study. Modern endocrine disruptor research now routinely examines mutile generational effects that were not considered in mid-century drug evaluation. The DES story illustrates that interventions acting on developing physiology can produce effects whose full scope appears only after an entire generation has passed. It also shows that absence of observed harm in short-term studies does not equal proof of long-term safety when the endpoint lies decades ahead. Finally, the episode underscores the value of retaining detailed exposure records. Without the maternal medication histories collected by Herbst's team, the causal link would have remained invisible. When new medical or policy tools are introduced at scale, what mechanisms exist to detect consequences that surface only in the next generation? That's unintended consequences for today. If this episode gave you something to think about, share it with a friend who'd appreciate the story. Subscribe wherever you listen, and we'll be back tomorrow with another case. And before you go, this show is part of the NARE Network, a family of daily podcasts covering tech, science, markets, and more. If you enjoyed today's episode, give SpaceX Daily a listen, the daily companion for following SpaceX as a public company from Starship to the stock ticker. You can explore the whole lineup at neurannetwork.com. This episode used AI voice synthesis of my voice. Editorial selection and analysis are my own.